Hello, ladies and gentlemen, and welcome to Mr. Saucedo's YouTube videos. We're going to be talking about RNA today. So we've spent a lot of time looking at DNA, and even at the very beginning of this unit, we were talking about how we weren't sure what was carrying genetic information, whether it was proteins, RNA, or DNA. So we might as well talk about RNA now, since we've been utilizing the term RNA and haven't really told you what it means yet. So first thing you need to know is that transcription is the process of making RNA from DNA. So let me pose a quandary to you. So DNA is the holder of all of our genetic information, okay? It is trapped inside of the nucleus, right? Nuclear pores only let certain things in, only let certain things out. So if the nucleus of the cell needs something done, how can it tell cell parts what to do? Well, if you're trapped in like prison, how could you go and send orders to other people? Well, you could write letters, right? You could send an email or something maybe. That would work. You could, in some way, send a message. That's like the point, right? So DNA will send messengers, okay? And those are that is what we would call RNA in order to manufacture things for us, in order to change things, in order to move things. All that's done via RNA. So RNA stands for ribonucleic acid, okay? So remember, DNA was deoxyribonucleic acid. RNA is ribonucleic acid. So how is RNA different from DNA? Well, first of all, it is single-stranded. So a lot of times people say it looks like a broken fence. It doesn't have a double helix shape. So that's the first one. Secondly, it uses ribose, not deoxyribose. Now, they're very similar to one another. Deoxyribose has like this little OH group here. Deoxyribose does not. And the most important one is RNA uses a nucleotide called uracil instead of thiamine. So instead of ATCG, we have AUCG for RNA. And so this is what a typical RNA molecule looks like. It looks something like this. So you can tell it's single-stranded. It has U's instead of T's. That's all you really need to be able to identify for RNA. So always remember, it's always AUCG. We do not have, though, okay, just to, again, double helixes. So we don't have double helixes. They're only single strands. Okay. Now, there are 3 types of RNA, but they all help to do the same kind of thing. So we have messenger RNA. They carry the blueprint for building something. So I always say that's kind of like the foreman of the building project, right? They have the plan, they have the blueprint, they know what's supposed to get made. Ribosomal RNA, otherwise known as rRNA, they're the actual workers that build the protein. So they do the actual work, though. And then tRNA, or transfer RNA, they're the ones that transfer amino acids to the protein. So those are like the tools. So each form of RNA has its own little job. One is the message itself. It is the blueprint. The other one has to build from that message. And then the last one needs to be the materials that are necessary in order to build the appropriate item. So how does transcription work? So the first thing that we need to do is to have the nucleus say, okay, great. I need you to build something for me. So when that happens, an enzyme called RNA polymerase is activated. RNA polymerase attaches to the DNA in a specific spot and begins to unwind and unzip the DNA. What does that sound like? Right? Sound familiar? It kind of should. It sounds a little bit like DNA replication. But notice, right, that so this is RNA polymerase. This is the piece of DNA. You can see it's unwinding part of the DNA right here, and you're getting a single strand off of this. So we're not making a copy of the DNA completely here. We're actually manufacturing a message that is contained in the DNA itself. Okay? So how does it know where to attach? That would be my first question. So it looks for a sequence called a promoter. Promoters signal where the RNA polymerase is supposed to attach. And so this is an example of a promoter. The most common promoter is called a TATA box. So it's a sequence of TATA on your chromosome somewhere. And so that is one of the most common promoters that lets RNA polymerase know, okay, I'm supposed to go there. Now, RNA polymerase creates a complementary strand, okay? So it's not, copying this exactly. Instead, it is making a message from it after the promoter. So the TATA box here is not included in the message, but always remember U is in the place of T. So right after the TATA box, I see an A. That means it would put a U there. C, that would be G, U, C, U. Now, how does it know where to stop? Once it hits a terminator, it detaches and transcription is complete. Now, and by the way, if you want to know what a good terminator is, it tends to be a long sequence of the same base, like just a bunch of T's in a row or something, and that'll let it know, okay, great. Okay, I don't need to translate that at this point. So to protect the mRNA, what do? This is a very, very important message. So what they do is it takes the message, it cleaves it off, it attaches a little cap to the top called a GTP cap, and it attaches a bunch of A's at the tail. So just kind of like we were talking before about telomeres at the end of our DNA, we want to protect the message itself. So instead, just adding a bunch of useless A's at the end of it, if something does happen, only A's will get lost and it won't be anything important instead. And again, that's the poly A tail. So if I give you this sequence of DNA that's moving 3 to 5, okay, and my promoter is AC and my terminator is TC, what would be my message? Okay, so first things first, AC is right here. That's the first AC I see. So I would just continue. Remember, A always goes with U, so we can fill in the rest. I stop where it says TC. So that would be my message. I would add GTP to that. I would add a poly A tail. I have my message now ready to get read by our rRNA. Okay, let's do another one. The promoter is TA, the terminator is CG. Right? That's the first TA I see, so I would continue. Okay, I would continue till I see the first CG, and then once I see the first CG, that's the end of my message. GTP, poly A tail. And just like before, this can happen extremely quickly.
It's unzipping the double helix and copying one of the two strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to copy the A's, C's, T's, and G's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related building block known as U. You are watching this process called transcription in real time. It's happening right now in almost every cell in your body.
And again, it stopped when it hit the appropriate terminator. All right, well, if you have any questions, let me know.